Chapter Body Fluid. Chapter Body Fluid. And circulation. This body fluid circulation, we will going to discuss the blood groups. Blood group is also called ABO blood types. ABO blood types. You see, in human population there are four major blood group: AB, A blood group, B blood group, O, and AB. Of these four, A, B, and O, it was first discovered by Carl Landsteiner. Carl Landsteiner was first discovered the ABO blood group. Who discovered there are two kinds of antigen. Carl Landsteiner was discovered there are two kinds of antigen on the surface of the RBC. You see, A, B, these are the two kinds of antigen usually present. That means what? Absence of this antigen. Antigen A, there are two antigen. You said no, there are antigen A and antigen B. These are the two antigen present on the surface of RBC. Present on the surface of RBC. Antigen A, antigen B present on the surface of RBC of human blood. Human blood. This antigen A, antigen B, which is present on the surface of RBC of human blood. Next you see this AB. This AB blood group, it was discovered by Dr. Alfred von de Castillo. This AB blood group, it was discovered by Dr. Alfred von de Castillo. This Alfred von de Castillo and Sterling. These two fellows are students of the Carl Landsteiner. Alfred von de Castillo and Sterling. These are the two fellows are the students of the Carl Landsteiner. Next one, based on the presence or absence, based on presence or absence of antigen. Based on presence or absence of antigen, there are three kinds of blood group. There are three kinds of blood group. There are three kinds of blood groups. What are they? A, B and O. Next one, see the blood group is the it is a type of inheritance. It is a type of blood group is the type of inheritance. Okay, blood group which is one of the type of inheritance. Okay, it is a type of inheritance. Blood group is the type of inheritance. Okay, you see this type of inheritance which is controlled by multiple allelism. Blood group system is one of the good example for the multiple allelism. Okay, usually see genes which consists of there are two alleles. Okay, but you see the multiple allelism which consists of more than two alleles. Okay, you see a gene which consists of more than two alleles. Okay, more than two alleles. Okay, more than two allele. Okay, you see a gene which usually consists of two allele which control a particular character, but you see the multiple alleles means what? Particular character is controlled by more than two allele. Okay, understand not? There are three autosomal allele which controlling particular blood group of any person, which is located in chromosome number 9. It's located in chromosome number 9. You see gene which controlling the blood group, gene which contains the allele. There are three autosomal allele which are located in a particular homologous chromosome in a particular locus, which controlling the particular blood group of any person, controlled by the autosomal allele which are located in chromosome number 9. You see gene which control the blood group, gene which control the blood group that we labeled as gene which control the blood group that we labeled as after discovered by the Carl Landsteiner, that we labeled as L, capital L. After that blood group that will be discovered by Landsteiner. Okay, listen not. Later on you see that L, capital L, that is the gene mentioned as capital L, labeled as capital L, after discovered by the Landsteiner, the blood group after discovered by the Landsteiner. You see after later on that the capital L that will be replaced by capital L or I from agglutination. Okay, that capital L now, due to the isoglycination, that will be named as a I. That gene will be now I. You see, I gene that exists in 3 allelic form. That I gene, okay, that is from isoglycination. That I gene exists in 3 allelic form. What are they? 3 allelic form. That is IA, IB and IO. Okay, this IA, IB, these two blood group IA, IB, okay, that I gene exists in 3 allelic form, that is IA, IB and IO. You see, IA and IB, these are all dominant blood group. Blood group IO is recessive form. Okay, let's see. Let us see what are the possible combination an individual can have out of these 3 allele. That means IA, IB and IO. Usually every individual could have only 2 allele. Okay, you see, let us see the combination. Usually there are 3 allele which controlling the blood group. Let us see the combination. IA, IB, IO. Next IB, next one IA, IB, IO, IA, IB, next IO, these are the combination. You see, IA, this blood group type is A. Blood group type is A. Next IA, IO, this also A blood group. IB, blood group B. IB, IO, blood group B. IA, IB, this is blood group A, B. IO, blood group O. Okay, you see, IA, this is what homozygous blood group A, but it is homozygous. Okay, this IA, IO allele is also possible for A blood group. This is the this is what heterozygous. This is what heterozygous. Okay. Next IB, blood group B, it is homozygous. It's also homozygous type. Okay, allele B, IA, IB. Okay, see next one IB, IO. This is what heterozygous, blood group B, but heterozygous. Okay, see next one IA, IB, blood group AB, but it is both A and B both are dominant. Okay, so it is called codominant type. IA, IO, which giving the blood group O, it is recessive. Okay, you see blood group A, B, AB and O. Okay, but you see genotypically there are six possible genotypes are came. Okay, 1 2 3 4 5 6 possible genotype came. Okay, due to these three allele there are 6 possible genotype. 1 2 3 4 5 6 possible genotype, 4 possible phenotype. That means what? Blood group A, B, AB and O. There are 4 possible phenotype that can be produced. Okay, let us see the individual blood group and to see which type of antigen antibody they possess. Let us see the blood group based on the presence of antigen antibody. You see blood group A. Blood group A means, you see genotype of this person. Genotype of this person, you see genotype of this person. See IA, blood group A means IA or IA and IO. You see this is genetic makeup. This is the genetic makeup IA or IO. So that allele. You see, as already we discussed that based on the presence of antigen antibody, you see blood group A, the antigen which is present on the surface of the RBC. This is the RBC. On the surface, you see antigen. This is a proteinous part. If blood group A means, that means you see the surface of the RBC, antigen is A. At the same time, blood group A person possessing the plasma of this person, you see antibody is B. Blood group B. Next one, blood group B means the genetic makeup, the allele formation. You see IB, but maybe IB and IO. Okay, this is what homozygous, this is what heterozygous form. Okay, genetic makeup, you see this homozygous IB, heterozygous form IB and IO. You see the surface of the RBC of this person possessing the proteinous part B antigen on its surface. But you see the plasma of the RBC, you see antibody A. Okay, you see next one, blood group AB. Next, blood group AB. Genetic makeup of this person, you see IA and IB. Okay, you see on the RBC surface there are 2 antigens. One is the A, another one is the B. These are the 2 protein spot antigens on surface of the RBC. but in his plasma does not possess any antibody. Okay, no antibody. Okay, these are the codominant allele. Okay, next one, blood group O. Next one, blood group O. In this blood group O, RBC surface no antigen. Okay, the RBC surface there is no antigen. Okay, this is what recessive allele. Okay, that's not IUO. Okay, these are recessive allele. See, RBC surface there is no antigen, but in his plasma of the blood possessing the both antibody. Okay, that's not plasma of this blood, you see possessing both antibody A and antibody B. Okay, but you see AB blood group, RBC surface possessing the two antigen A and B, but there is no antibody. Here is the blood group O. There is no antigen on the surface of the RBC, but possessing the A and B antibody.